Hello everyone. We will discuss about the most important topic in the biotechnology chapter in the 12th class biology, that is gene therapy topic. Gene therapy. Gene therapy. It is a method to correct a gene defect. It is a method to correct a gene defect. If there is any gene which is defected, we can correct the defected gene with the help of this method, and properly it has to be diagnosed first in the child or embryo. Okay, so after diagnosis only, after diagnosis in the child or embryo, in the child or embryo itself. Okay, next, here the genes are inserted into a person's cell. In this concept, in this method, the genes are inserted into the person's cell, person's cells or tissues. Here the genes are inserted into the person's cells or tissues to treat any particular disease, to treat any particular disease. So in this, it will compensate for the non-functional genes. These genes are going to compensate the non-functional genes. We are doing this to compensate the non-functional genes. We are doing this. So this is called gene therapy, and we are going to study one example for this. So this example is about the first clinical gene therapy. The first clinical gene therapy. The first clinical gene therapy was done in the year of 1990. In the year of 1990. To a 4-year girl. Her name is Ashanti De Silva. By a doctor called James Anderson. Dr. James Anderson. And she was suffering from, this 4-year girl, Ashanti De Silva, she was suffering from a disease called adenosine deaminase deficiency. She was suffering from adenosine deaminase deficiency. Adenosine deaminase deficiency. Adenosine deaminase. It's an enzyme actually. This enzyme is useful in immunological reactions. It will induces immunological reactions. Generally gets synthesized from the bone marrow. Due to the deficiency of this particular enzyme, it may leads to severe combined immuno disorder. It's SCID. SCID. It's commonly we call it SCID. Severe combined immunodeficiency. Here, very simple thing. Bone marrow in the from the bone marrow. There are particular in the genes. That gene is nothing but ADA gene, which is responsible for the ADA protein, and thereby this protein will be transformed into ADA enzyme. ADA enzyme. In the due to the defective gene, okay, due to defective ADA gene, this ADA enzyme will not be synthesized. Will not be synthesized. So it leads to the deficiency of ADA. ADA deficiency. Why? Because this is defective gene. If the ADA gene is defective, this ADA enzyme won't be synthesized, and it may leads to the disorder called severe combined immunodeficiency. Severe combined immunodeficiency. Now, what are the treatments that we have? What are the treatment options that we have? Generally, bone marrow transplantations we have. Treatments. There are two treatments. Number one, bone marrow transplantation. Bone marrow transplantations. Bone marrow transplantation. Bone marrow transplantation, or else enzyme replacement therapy. Enzyme replacement therapy. Enzyme replacement therapy. So these are the two treatments which were available, but these approaches are not completely curative. The thing is not completely curative. Not completely curative. They are not completely curative. Then what to do? Even though bone marrow, you may get a question, sir, what about bone marrow transplantation? Bone marrow itself changed, no? But there might be some incompatibility issues arises. There might be some incompatible issues may arises in the recipient's body, okay, after some time. On the long run, it may not be effective. And enzyme replacement means every time the enzyme has to be replaced. So it is very difficult. So there is a technique, there is a method called gene therapy. There is a method called gene therapy. So this gene therapy is nothing but the treatment of defective ADA gene with the normal functional ADA gene. Very simple. From the patient's body, the lymphocytes are collected. The lymphocytes are collected. Lymphocytes are collected. Lymphocytes, we already know that lymphocytes are the types of WBC. After collecting lymphocytes from the patient's blood, they have to be grown in the culture. They have to be grown in the culture. Proper culture we have to provide, and we have to grow these WBC lymphocytes. We have to grow, okay, grow in the culture. After growing in the culture, we have to transfer the functional ADA cDNA. This is nothing but a functional gene for ADA gene, functional ADA gene, ADA cDNA. This is a gene. By using a retroviral vector, okay, retroviral vector. We have to use some vector to transfer DNA, right? Anyhow, so that vector here is retrovirus. Okay, some virus which can perform reverse transcription of its RNA to form DNA. So that kind of virus we are selecting as a vector to transfer this ADA cDNA into the patient's lymphocytes. Patient's lymphocytes. And after that, we have to transfer to the patient's body. So this has to be periodically repeated. It has to be repeated actually. Why? Because the lymphocytes will be having very least lifespan, okay, 1 to 2 weeks only, right? The lifespan of RBC is only 2 weeks, right? So it has to be repeated further. It has to be repeated further. But the permanent thing is diagnosis of this ADA deficiency, diagnosis of ADA deficiency in embryonic stages itself. We can do that in embryonic stages only. If you are going to treat ADA gene, definitely from the bone marrow cells, okay, ADA deficiency, it should be diagnosed in embryonic stages itself. Whenever you are going to transfer this normal ADA cDNA gene into the bone marrow cells in embryonic stages, it could be give a permanent cure. It could be given a permanent cure. That's it. So this is all about the gene therapy. Hope you understood. Thank you very much.